Hi all. How well do you know bacterial identification techniques like biochemical tests and antibiotic sensitivity testing? Challenge yourself with this microbiology quiz and test your knowledge. This quiz covers bacterial identification methods like biochemical tests and antibiotic sensitivity test. Like, share and subscribe for more microbiology quizzes and study tips, and turn on notifications to stay updated. So the first question is, for identification of gram-negative bacteria, the common biochemical tests used are abbreviated as ICUT, which stands for all the following except. So the abbreviation ICUT stands for common biochemical tests used for gram-negative bacteria, which are indole test, coagulase test, urease test, triple sugar ion test. One of this is not included as in ICUT. So which one is not included in ICUT? The answer is coagulase test. So coagulase test detects the presence of coagulase enzyme which clots blood plasma. It is used to identify Staphylococcus aureus, a gram-positive organism which produces coagulase. So it is not used in ICUT, that is gram-negative bacteria cannot be identified by coagulase test. So coagulase test is a test used for Staphylococcus aureus, which is tested by the clotting of blood plasma. So if there is a clot formation, it is called as coagulase positive. It can be done on a slide and also on a tube. So in clot formation if occurs in this tube, it is called as coagulase positive. So it is Staphylococcus aureus. Then what is this ICUT? It is indole, citrate, urease and TSI test. It is not coagulase. C stands for citrate. What is indole test? Indole test checks if a bacteria can break down tryptophan amino acid to produce indole. So if there is indole produced, there will be a red ring. This is indole negative. There is no red ring present. What about citrate test? Citrate test determines if a bacteria can use citrate as a sole source of carbon. So this is a negative citrate test, and if it is positive, citrate is utilized, it will be Prussian blue color. What about the third one, urease test? This identifies bacteria which can produce urease. Urease enzyme breaks down urea into ammonia. So there will be a pink color formation in urease test, which is called as urease positive. What about TSI? TSI is triple sugar ion test. It evaluates a bacteria's ability to ferment glucose, lactose and sucrose, as well as its ability to produce hydrogen sulfide. So this TSI, triple sugar ion agar, if this has red and red, that is here it is red and red, there is no sugar fermentation. This is used as control. This is red and yellow color. Glucose is fermented, but lactose and sucrose not fermented here. Yellow and yellow, that is glucose will be fermented, lactose or sucrose fermented, and here there is black coloration, that means H2S is produced. So the next question is, triple sugar ion medium contains the following sugars except. Is it sucrose, glucose, lactose or maltose? Yes, sure. You know the answer. The answer is maltose. So TSI is used for testing of acid and gas production from fermentation of glucose for production of hydrogen sulfide. So this is triple sugar ion slant. So if it ferments glucose and ferments lactose and sucrose, there will be yellow color formation. And if it does not ferment, there will be only using peptone, there will be no color change. If it runs out of glucose and cannot use lactose, sucrose and uses peptone, there will be yellow color in the butt and red color on the slant. And if there is H2S production, black color also will be there. This is how triple sugar ion test is interpreted. Now the next question is, which of the following uses both diffusion and dilution methods for antimicrobial susceptibility test? Antimicrobial susceptibility test, or AST, it helps physicians to determine the appropriate antimicrobial for treatment. So when the infection occurs, patient sample will be taken and empiric therapy will be started. Then in the laboratory, AST will be conducted on the sample, that is antibiotic susceptibility testing. And according to its report, therapy will be adjusted based on that test result. Then the patient responds positively to the treatment. So which of the following uses both diffusion method and dilution method for antimicrobial susceptibility testing? Is it Kirby-Boer method, Stokes method, broth dilution method, or epsilometer method? The answer is epsilometer method. It is also called as E-test. It determines the minimum inhibitory concentration, or MIC, of antibiotics against bacteria and fungi. So it is a quantitative method as well that uses a plastic strip with a gradient of antibiotics. So here it is the gradient of antibiotics, and when it is placed on the bacterial inoculum, after overnight incubation, you can see a zone of inhibition like this. So this point indicates the minimum inhibitory concentration. So the antibiotic on this strip will be diffused first, and here it is diluted also. So this method uses both diffusion method and dilution method. Whereas Kirby-Boer method is a disc diffusion test. It only uses diffusion method. It determines how sensitive bacteria are to antibiotics and other antimicrobial compounds. So here this plate is inoculated with bacterial solution or suspension. Then antibiotic discs are placed on over the surface. After incubation, overnight incubation, you can measure the zone of inhibition. So this is the antimicrobial disc and this is the zone of inhibition. You can see bacterial growth here. So this zone of inhibition is measured. What about Stokes method? It compares the zone sizes of test and control organism to determine antibiotic susceptibility. So in this portion there is control strain inoculated, here also control strain, and in this middle portion there is test strain inoculated, or you can do it otherwise also, that is control strain in the middle and test strain in the both these portions. So here you can see a clear comparison between the test strain and the control strain. Here both test and control gives the same diameter, here also, and here you can compare, is it larger or smaller zone of inhibition. And broth dilution method determines how susceptible a microorganism is to an antimicrobial agent. Here dilution of antibiotic solution is done in which the organisms are inoculated. Here only dilution is done. Next question is, what should be the minimum distance between two adjacent antimicrobial discs on the Muller-Hinton-Agar plate? Muller-Hinton-Agar plate is used for Kirby-Boer method of disc diffusion test. And the question is, what should be the minimum distance between two adjacent antimicrobial discs on the Muller-Hinton-Agar plate? Is it 55 millimeter, 40 millimeter, 32 millimeter, or 24 millimeter? The answer is 24 millimeter. That is, the minimum distance between the two discs should be 24 millimeter. It is to prevent interference between the diffusion zones of different antibiotics after overnight incubation. And this is typically achieved by ensuring a consistent spacing across the plate. And the distance between the plate and the disc also should be at least 10 to 15 millimeter. Next question is, all the following automated systems can perform antimicrobial susceptibility testing except Vitek2 system, MALDI-TOF system, Phoenix system, or microscan walkaway system. The answer is MALDI-TOF system. MALDI-TOF or matrix assisted laser desorption ionization time of light. Mass spectrometry. It uses a laser to identify bacteria, fungi, and viruses, but it cannot determine antimicrobial susceptibility testing. So here sample is applied to target. It is overloaded with matrix solution. Then laser is ported on the target. Then there will be desorption ionization molecules, desorbed ionized molecules, which will be accelerated by the potential differences through a high vacuum and field free flight tube. And the time of flight based on mass of particles is captured on this detector, and the resulting spectrum is compared to library which contains the patterns of clinically relevant species. That is how MALDI-TOF identifies the organism. Vitek2 is a fully automated system which identifies bacteria and tests their susceptibility to antibiotics. It uses fluorescence-based technology, so it can detect the antimicrobial susceptibility as well. Phoenix system. It also is an automated microbiology system that identifies bacteria and tests their susceptibility to antimicrobial agents. Microscan walkaway system also used for identification and antimicrobial susceptibility testing. So to summarize, you now know what are biochemical tests. These tests are used in microbiology to identify microorganisms based on their biochemical properties. And how antibiotic sensitivity test is done, which is done by taking a sample from the infected area of the body and growing bacteria from that sample, that is clinical sample, and then exposing the bacteria to the antibiotics. Thank you.